I'd like you to begin by reviewing the learning outcomes for this course as presented on the first day and summarized here. I will be presenting a lot of material over the next 5 lectures, much of which builds upon foundational knowledge that you may have encountered previously in biology and chemistry and perhaps other courses. It may be useful to refer to a basic biology textbook, and many of the figures we use in this course are drawn from Campbell Biology, which therefore provides a useful reference if you'd like more information. I will generally include on my slides references to the sources of the material I am using, and will include on the topic summary sheet recommended readings to help deepen your understanding of the material. The learning objectives for today's lecture are membranes. We want to consider how membranes form, and specifically this requires that we consider the role of water, the solvent of life, and how the hydrophobic effect leads to the spontaneous self-assembly of biological membranes from their component molecules, often phospholipids. We hope that you will be able to describe membrane bilayer organization and understand the fluid mosaic model for membranes, how they're assembled and how they function. Further, we'd like you to understand how lipid composition, changes to the molecules that make up the membrane, can affect fluidity and integrity of membranes. And we'll conclude with a discussion of how membrane proteins are inserted into and interact with the membrane. In the first part of this course, we will focus on cellular physiology. Cell physiology addresses the questions of how individual cells meet their nutritional needs. How do they grow and divide? How do they respond to a changing environment? Later in this course, we'll shift our focus to multicellular organisms and organismal physiology. In this case, we'll be considering how cells come together to function in the context of complex organisms through the formation of tissues, organs, and organ systems. And we'll learn about systems such as the circulatory and respiratory systems. Many of the systems in multicellular organisms meet the same functions as those we will consider at the level of individual cells: how to grow, obtain nutrients, dispose of waste, how to exchange gases, and how to reproduce. All life is based on cells. The idea that life is based on discrete units known as cells is a foundational concept in biology. For multicellular forms of life, plants and animals, with which you're probably most familiar, many cells come together to form an organism, and many specialized cell types work together as tissues and organs to provide for a living, functioning organism. However, as this phylogenetic tree of life illustrates, based on the true genetic diversity found amongst living organisms, animals and plants are really a small corner of the phylogenetic tree, and much of the vast diversity of life on Earth is invisible to the naked eye. We have the bacteria and the archaea and many unicellular eukaryotes that are all part of this grand tree of life. And we'll learn more about these different organisms throughout this course. A direct comparison of a eukaryotic cell, such as an animal or a plant cell, and a bacterial or prokaryotic cell is shown in this figure from Campbell Biology. Here we see a very simple bacterial cell containing DNA and surrounded by a membrane, and a much larger eukaryotic cell also surrounded by a membrane and having internal compartments separated by membranes, such as the nucleus and membrane-enclosed organelles. In all cell types, the boundary of the cell, the layer that separates the cell from the environment, is known as the cell membrane, sometimes called the plasma membrane. All free-living cells have DNA as their genetic material, ribosomes for protein synthesis, and much of the biochemistry that occurs in cells has been conserved across life, which is one reason scientists believe that life evolved and diversified from a single ancestor, a last universal common ancestor, as shown in the phylogenetic tree mentioned earlier. Let's consider then the cell or the plasma membrane as the key boundary layer that functions to separate the living cell from its surroundings. And this is true for single-celled organisms, and these plasma membranes also separate discrete cells in multicellular organisms. So shown here, we can see this membrane as a lipid bilayer, and we will consider what that means in subsequent slides. And within this lipid bilayer, there are these purple blobs, as shown here, which are proteins. These represent proteins spanning or sitting on the membrane. On the inside of the cell, we have all of the cellular complexity of the DNA and RNA and proteins and metabolites that make up the cytoplasm. And outside the cell, in this example, there are extracellular matrix, filaments, and other macromolecules. The major learning objectives of our talk today are to be able to explain the role of water as the solvent of life and how the hydrophobic effect leads to the self-assembly of biological membranes, and then we will turn our attention to membrane bilayer organization, the fluid mosaic model, more details of lipid composition, including both lipids and proteins. Beginning then with water. Water is truly an amazing molecule. It is, of course, familiar to all of us. The biosphere contains abundant water. It's considered the solvent of life. Water on Earth exists in all three physical phases. It exists as a solid, ice, a liquid in the oceans and lakes and in our bodies, and as a gas in the biosphere. Why does water have the unique ability to exist in all of these states under physiologically relevant conditions? The properties of water derive from its very simple structure, H2O, and the fact that the oxygen atom is much more electronegative than the hydrogen atom. So when two hydrogen atoms are bound to oxygen, the electron, normally associated with hydrogen, spends more of its time near the oxygen nucleus than it does the hydrogen nucleus. And this leads to a partial positive charge exposed where the hydrogen atom is, a proton, and the oxygen has an excess or an accumulation of negative charges. These partial charges are given the symbol delta, lowercase delta, delta plus and delta minus. These neighboring molecules then attract each other because opposite charges attract. And this is referred to as a hydrogen bond. Hydrogen bonds are extremely important in biology, and I encourage you, if you're not familiar with hydrogen bonds, to explore some of the readings that discuss this concept. Water is an excellent solvent. Here on the left, again, these are figures from Campbell Biology. We can see that because of its polarity, having both negatively charged oxygen atoms with partial negative charges and protons or hydrogen atoms with partial positive charges, water can easily coalesce around positive ions such as sodium and negative ion, shown here chloride, and thereby dissolve salt. So NaCl crystals or table salt will dissolve in water, as I'm sure you're all aware. Similarly, many biological macromolecules, large molecules important in biological systems, have on their surface charges. And these may be full charges or partial charges, and they can be well solubilized in an aqueous environment. So proteins, carbohydrates, DNA and RNA are all water-soluble macromolecules, and water is an excellent solvent for these polar macromolecules. Water is polar, and it is good at dissolving polar macromolecules. I'd like to first briefly review the chemistry of life, because this will help us to understand the unique properties of lipids, which comprise membranes. Much of biology focuses on macromolecules. Macromolecules are often long polymers made up of individual units called monomers. So many of the most familiar molecules in biology are polymers. This includes carbohydrates, sometimes called polysaccharides, many sugars. So carbohydrates are polymers. Proteins are polymers made up of amino acids. They're sometimes called polypeptides, peptide being another word for amino acids or collections of amino acids. Nucleic acids are also polymers made up of nucleotides. This is the GATC of the genetic code in the case of DNA. Unlike these macromolecules, lipids are moderate-sized molecules that are not really made up and assembled of individual monomers in the same way. But lipids are important macromolecules that can assemble spontaneously together to make membranes, and we'll consider how that occurs. So to consider how it is that lipids assemble into membranes, we need to briefly review the types of bonds that occur in chemistry. So molecules are collections of atoms held together by covalent bonds. Covalent bonds are very strong bonds. But biology, much of biology involves interactions that are much weaker. We consider these as to be much weaker bonds, and they break and form on a much more rapid time scale. Molecules themselves may persist for many hours or generations, whereas weak bonds form and break on a time scale of seconds or even nanoseconds. So hydrogen bonds, as I mentioned previously, are an example of a weak bond. Bonds between charged atoms, ionic bonds, are considered weak, as are van der Waals forces, which contribute importantly to the hydrophobic effect. Van der Waals forces are weak interactions between uncharged molecules due largely to transient dipoles that form in the atomic components. To a first approximation, the cell membrane can be considered as a phospholipid bilayer. The membrane actually has about an equal mass of lipids, shown here in green and purple, and proteins, shown here studded in the membrane. The phospholipids form a bilayer because they are amphipathic. They contain both hydrophobic and hydrophilic regions that allow them to self-assemble as a stable boundary that can thereby separate the two aqueous compartments, the inside of the cell or cytoplasm, and the external space. What do we mean by amphipathic molecules? Phospholipids that comprise the bilayer have a hydrophilic head group and a hydrophobic tail, which is made up of molecules known as fatty acids. And these two groups have different properties, hence amphipathic, both tendencies. The head groups often are charged or polar. In this case, there is a phosphate, which has a negative charge on the oxygen, and a quaternary amine, a nitrogen atom with 4 bonds, and which carries a positive charge. So this is a polar head group that wants to be solvated by water. So that is hydrophilic or water-loving. These hydrophobic tails, on the other hand, are nonpolar, hydrophobic or water-fearing and want to be sequestered away from water, so they cluster together on the interior and help make the bilayer membrane. So why do these fatty acid tails, shown here, pack away from water, exclude water? This is an example of the hydrophobic effect and requires that we revisit the properties of water. So water, as we've already mentioned, loves to interact with polar molecules, including itself through the formation of extensive hydrogen-bonded networks. In the solid phase of ice, these molecules crystallize in place in these hexagonal arrays, which is why snowflakes have 6 sides. In liquid, it assumes a similar structure with many hydrogen bonds, but the water molecules are constantly moving, breaking and reforming hydrogen bonds millions, even billions of times per second. This is the very dynamic structure that allows water to be such a good solvent. If, however, a hydrophobic molecule is placed in water, the water molecules are unable to form favorable hydrogen bonding or polar interactions with these hydrophobic carbon atoms, carbon chains, and instead they form a structure, a cage-like structure, sometimes called a clathrate, of water molecules around the hydrophobic tail. And this is unfavorable energetically because it minimizes the mobility of these water molecules. It restricts their ability to hydrogen bond with other water molecules and to move around freely in solution. So there's actually an entropic cost to the insertion of a hydrophobic alkyl group into water. The hydrophobic effect describes how hydrophobic molecules tend to self-associate to minimize this energetic cost. One important family of hydrophobic molecules in biology are the fats, sometimes also known as oils if they are liquid at room temperature. So on the left here, we see the structure of the components that go into building a fat. On the one hand, we have this long hydrophobic alkyl chain. This is a saturated carbon chain, very simple carbon chain. Here shown is palmitic acid. On the left, there is a carboxylic acid, a C double bond O COH acid function. This is considered a functional group in chemistry. To the left of that, we see the simple 3-carbon compound glycerol. It's considered a polyol because it has 3 OH functional groups or alcohols. So this is glycerol, a very important 3-carbon compound in biology. When fatty acids are linked to glycerol by removal of water, we consider this an example of dehydration synthesis. The bond that is formed is considered an ester linkage. Ester linkages are quite common in biology. And because this glycerol has 3 alcohol groups, 3 OH groups that can each be esterified with a fatty acid, 3 different fatty acids can be linked to glycerol. We call this a triglyceride or a triacylglyceride, commonly known as a fat. So fats play many important roles in biology. Fats characteristically do not mix well with water. In the human body, they form droplets that are accumulated in specialized cells called adipocytes, which are found underneath our skin and throughout tissues. And they play an important role as a storage form of energy. Fats are a very high energy compound, and they're a way of storing energy. An example of a fat molecule in this space-filling representation is shown here with the polar groups, red being the oxygen, and these long hydrophobic side chains. Remember that in the case of an aqueous environment, these hydrophobic side chains would want to pack closely against each other. This illustrates the diversity of different polar head groups that are seen in membrane lipids. So on the right side here, we see phosphatidylcholine, phosphatidylserine, phosphatidylglycerol, typical membrane phospholipids. The bottom part of each of these phospholipids, as mentioned previously, is a fatty acid. Examples of fatty acids are shown on the left here. Free fatty acids are not a major part of membranes, but these are the precursors that go into building the membrane phospholipids. So cells can vary the properties of their membranes, their lipid bilayers, in two ways. They can change the type of polar head group from choline to serine to glycerol to ethanolamine, and they can insert other hydrophobic compounds into the membrane. So by varying the head groups, they can change the charge and properties of the cell membrane, and they can also change the length and the shape of the fatty acids that make up these phospholipids. Both of these types of changes are important in biological membranes. Next, I want to turn from our discussion of fats back to consider the membrane lipid bilayer and delve deeper into this concept of a fluid mosaic. So membranes are considered to form a fluid mosaic, as I've already introduced, where we have a phospholipid bilayer and spanning the bilayer or next to the bilayer, often proteins. So these phospholipids are not covalently linked to one another. They are held together only by these weak hydrophobic forces. So these phospholipids can diffuse around in the plane of the membrane from side to side. And that's why it's considered a fluid mosaic. The membrane needs to be fluid so things can move around. The proteins float in the membrane, held here by their own hydrophobic interactions, as we'll see, and they too can float from side to side, so they have fluidity. The ability of proteins to move depends on the fluidity. So the phospholipids that make up this bilayer spontaneously assemble. They can freely diffuse side to side, as can the membranes, but the charged parts of the proteins and the charged parts of these hydrophilic head groups cannot easily cross the bilayer. So the phospholipids don't flip from side to side with very high frequency. Instead, they move laterally. It turns out that maintaining proper membrane fluidity is essential for life. So we need to have the ability of these phospholipids and other membrane components to move, to slide past one another and have fluidity so that the proteins that are anchored in the membrane can also move from side to side. So many proteins diffuse from side to side along the plane of the membrane unless they are anchored to extracellular structures or to other adjoining cells. To visualize how proteins move along cell surfaces, scientists use engineering techniques to generate forms of the membrane proteins that are fluorescent, where they can be visualized as these light spots. In this case, proteins are visualized on the surface of a bacterial cell as it grows, and these proteins move rapidly from side to side. Some proteins diffuse randomly around the surface of the membrane, and others have a directional motion, in many cases powered by ATP. So these complexes are actually involved in building the bacterial cell wall and move laterally around the cell as it grows. In addition to the plasma membrane, the outer or cell membrane, eukaryotic cells have complex intracellular membrane systems. And these are important in their own right, but they're also important for the generation of the plasma membrane. So membrane proteins destined to sit in the plasma membrane, such as the G protein coupled receptor, are synthesized in the cytoplasm on ribosomes that will dock to the surface of the endoplasmic reticulum, where they will be inserted into the membrane. They will be carried in vesicles from the ER, fused into the Golgi apparatus, where the proteins may be modified, for example, through glycosylation, and then from the Golgi carried by yet a further set of vesicles that fuse to the cell surface. And this secretory pathway allows proteins synthesized at the ER to be inserted into the plasma membrane, or if they're destined to be secreted into the environment. In general, bacterial cells have limited or very few intracellular membranes, although there are exceptions. Shown on the right here is a photosynthetic bacterium where you can see that when it is active in producing photosynthetic complexes, it accumulates a large amount of intracellular membrane. Next, I want to turn to the question of how cells can vary their membrane lipid composition to control the fluidity, to be sure that the lipids and proteins can move around in the membrane to support life functions. Let's consider first the key role of the fatty acid chains that make up membranes. So I'm going to switch back now from talking about phospholipids, which are diacylglycerols, return to the molecules I introduced previously known as fats. So fats, you'll recall from earlier, are triacylglycerides. So they have the glycerol, this 3-carbon compound with 3 OHs, now linked by an ester linkage to long hydrophobic chains. So if you have saturated fats, such as found in butter, for example, these long, straight chains pack closely together and are strongly interacting with each other through the hydrophobic effect, through weak van der Waals interactions, and they form the solid butter, which of course, if you were to warm it up, it will melt. On the other hand, this type of fat or oil has a double bond, a cis double bond. It's called a monounsaturated fat, and it tends to be liquid at room temperature. Fats that are liquid at room temperature we call oils. So the example here is olive oil. And these are both discussed in Campbell Biology, and you can read about lipids and fats and oils in Wikipedia and many other sources. To give a little more detail, I looked up the composition of these fatty acyl chains as typical. Cells from all domains of life actively regulate membrane fluidity. They have to maintain the membrane fluidity within a healthy range. Viscosity is another name for fluidity, so this process is sometimes called homeoviscous adaptation. So cells will change the shape of their fatty acids depending on the temperature in which they are grown. So as a first example, I show here some old data from studies of the model bacterium E. coli, in which E. coli was grown either at very low temperature or at much higher temperatures, near body temperature, 37 degrees C, for example. And what you can see is as E. coli is shifted from 37 degrees, where it has mostly saturated fatty acids in its lipids, to much lower temperatures, the number of saturated fatty acyl chains goes way down, and double bonds are introduced to a much higher level. So by putting kinked fatty acid chains into the membrane, it helps that membrane to remain fluid. It helps that membrane to retain the fluidity properties we think of when we think of olive oil and not allow the cell membrane to turn into a stick of butter. So of course these phospholipids have only 2 acyl chains, unlike the 3 in fats, but the properties are similar. This is not unique to E. coli, of course. Maintaining proper membrane fluidity is also critical for animal cells. In the case of animal cells, cholesterol plays this role, At high temperatures, cholesterol prevents excess fluidity. Conversely, at low temperature, cholesterol helps maintain fluidity. So cholesterol is a bit of a puzzle because it acts as a fluidity buffer. At high temperatures, it helps limit the membrane from being too fluid by inserting here into this membrane. And at low temperatures, it helps it maintain fluidity. Cholesterol is essential for animal cells. It comprises up to 30% of the lipids in animal cell membranes. It's produced by the liver and, of course, also consumed in our diets. Because it's hydrophobic, this molecule doesn't readily diffuse around our bodies in a soluble form, so it's carried in the blood associated to proteins, lipoproteins. And for those worried about their cholesterol, doctors will measure blood level cholesterol, and they'll measure the levels of high-density lipoproteins and low-density lipoproteins. Too much cholesterol, as well as fats, triacylglycerides, are well known as risk factors for atherosclerosis, in which these insoluble hydrophobic molecules form aggregates known as plaque in arteries, which can lead to heart attacks. The final objective for today is to discuss a little bit about the membranes that function within the lipid bilayer and specifically how these membranes interact with the bilayer. So membrane proteins are categorized according to how they interact with the membrane. An integral membrane protein is one that extends through the bilayer, often with amino acids and surfaces exposed both on the inside of the cell and the outside of the cell. Peripheral membrane proteins are those that may associate with the surface of the membrane but not extend into the membrane or through the membrane. Proteins that interact with the membrane have to have regions that are compatible with the hydrophobic region of the fatty acyl chains. So a typical protein shown here, a soluble protein, is highly charged and polar. So proteins often have polar groups exposed on their surface, and this can be visualized in a space-filling model of a protein as shown here, where red indicates the presence of negatively charged amino acids, blue, positively charged amino acids, and white are those regions of the protein surface where there are exposed hydrophobic groups. For a protein like this, a typical globular protein that might be found in the cytoplasm, you can see that it would be very difficult for a protein like this to insert into the membrane. All of these hydrophilic amino acids want to be hydrated. They want to be solubilized and interacting with water, whereas they would not mix well with the hydrophobic fatty acyl chains in the lipid bilayer. Remember, hydrophilic and charged and polar groups like to interact with other polar groups. Hydrophobic groups like to interact with other hydrophobic groups. If we instead consider a transmembrane protein, we see that it has a very different type of charge distribution. In this case, the leucine transporter from bacteria, you can see that on the external surface of the membrane, there are many red groups, negatively charged. On the inner surface, there are many blue and some red groups, again charged. And if we were to look at the central region here, there's a band of non-polar surface. And that is perfect for allowing this protein to sit comfortably within the membrane. You can see here that this protein represents a sort of globular shape that spans the membrane. We would therefore categorize this as an integral membrane protein. And if you remember details of how proteins fold, you'll notice that many of these transmembrane segments are alpha helices, a very common secondary motif in proteins. How do these proteins get into the membrane? Membranes play a variety of physiological roles in the cell. Some of the major roles are listed here. These include the transport of water and solutes, a major topic for today, as well as cell-cell recognition, joining of adjacent cells in multicellular organisms, such as animals and plants, attachment of cells to their cytoskeleton or the extracellular matrix, enzymatic activity, and signal transduction. These topics are discussed in further detail in Campbell Chapter 7 for those who seek further reading. Proteins determine many of the functions of the membrane, including transport. Transport can involve water, shown here as these blue spheres, moving across a membrane. Although water can transit across membranes in an uncatalyzed reaction, the presence of membrane transporters for water can increase the flux. For many compounds, transport across the membrane absolutely requires proteins that allow either the facilitated transport down a concentration gradient or with the use of energy, such as ATP, can allow molecules to be transported up a concentration gradient. Cell-cell recognition is a second function of membrane proteins. Shown here is two eukaryotic cells closely juxtaposed, with one having an integral membrane protein modified with carbohydrates, chains of sugars, as a ligand or a binding partner for a second cell, which also has an integral membrane protein, with a pocket that is complementary to the carbohydrate side chain of the first protein. So this type of intermolecular recognition helps cells adhere to each other and recognize each other. Proteins also function in intercellular joining. In vertebrates, including humans, many cells are joined by what are called gap junctions, made up by a protein called connexin, whereas in plants, joining between cells and the exchange of solutes occurs through plasmodesmata. So proteins can play important functions in helping cells join and communicate with each other. The next function I want to discuss is attachment of a cell, again a eukaryotic cell, to the cytoskeleton or extracellular matrix. So we focused so far mostly on the structure and function of this cell or plasma membrane, but many eukaryotic cells are surrounded by a complex environment, which may include an extracellular matrix. This matrix may include a variety of polymers, proteins and carbohydrates, shown here as collagen, fibronectin, and this matrix provides a substrate for the interaction of many different cell types through transmembrane proteins. Proteins are also extremely important in cell signaling. So many proteins have an enzymatic function. They may function as enzymes in a variety of ways, and some of these enzymes are important in signal transduction. So these are two additional important functions of cell membrane proteins. Shown here is an integral membrane protein representative of a large family of proteins in humans and eukaryotes, the G protein coupled receptors, GPCRs. These proteins sit within the membrane and respond to ligands or signaling molecules from the environment. As their name indicates, these G protein coupled receptors interact with another protein, a G protein. G proteins are so named because they can bind either GTP or GDP. Upon activation, the G protein becomes loaded with GTP, and this activated form of the G protein then initiates downstream signaling events, which may involve the interaction with other membrane proteins to initiate a cellular response. So membrane proteins with carbohydrate modifications are also important in host pathogen interactions. Here we see a virus. This is the flu virus particle. This is an RNA virus, and it's a member of a class of viruses called enveloped viruses, which means they are packaged in a lipid bilayer surrounded by proteins. In the case of the flu virus, two major surface proteins on this viral particle are the hemagglutinin and the neuraminidase. The neuraminidase binds to sialic acid, carbohydrates, on the surface of human cells and allows the process of infection to begin. And these different proteins on the surface of the flu virus, the hemagglutinin and neuraminidase, are antigens for our immune system. So in this case, this is an H5N1 flu virus, representing that it has the hemagglutinin type 5 and the neuraminidase type 1. And these vary from virus to virus, which is why we often need to get a different flu shot each season. To review then, today's discussion of membranes centered around the following learning objectives, which are summarized again here. I encourage you to review the chapter in Campbell Biology, Chapter 7, in which membranes are discussed in some detail. I also refer you again to the topic summary sheet and suggest that you consider reviewing Chapters 2 through 5 in Campbell Biology to refresh your background on some of the chemical and foundational concepts that will be helpful as we continue our discussion of cellular physiology.